Good morning, sir. Today, I, Achal Bora of MSc Microbiology, second semester, is going to deliver my presentation on the topic of fermentation by Enterobacteriaceae and Clostridia. These are my content. First of all, introduction to Enterobacteriaceae. The family Enterobacteriaceae is part of the domain Bacteria, phylum Proteobacteria, class Gamma Proteobacteria, and order Enterobacteriales. They include bacteria that naturally inhabit the mammalian gut but also can occur and multiply in other environments. For example, species of Escherichia, Citrobacter, Enterobacter, Proteus, Hafnia, Klebsiella, Providentia, and Serratia. Here is a label diagram of Enterobacteriaceae. Now, characters of Enterobacteriaceae. All Enterobacteriaceae are large heterogeneous group of gram-negative rods. All members of the Enterobacteriaceae family ferment glucose with acid production and reduce nitrates into nitrites. Enterobacteriaceae are typically oxidase negative, meaning they either do not use oxygen as an electron acceptor in the electron transport chain or they use a different cytochrome enzyme to transfer electrons to oxygen. Nearly all are facultative anaerobes. They are motile with the exception of Shigella and Klebsiella, which are non-motile. These bacteria have peritrichous flagella. Enterobacteriaceae are usually non-capsulated except Klebsiella pneumoniae or simply as Klebsiella, which is a gram-negative capsulated non-motile rod-shaped bacteria. Enterobacteriaceae are non-fastidious bacteria that do not need such special nutritional supplements or conditions. They can grow well on bile-containing media, that is Macunkie agar. Here is the image shows Shigella, Proteus and Escherichia growth on Macunkie agar, whereas this picture shows Yersinia growth on Macunkie agar. Now, basic classification of Enterobacteriaceae. First, lactose fermenters, for example, E. coli and Klebsiella. Second, late lactose fermenters, for example, Shigella shunai. Third, non-lactose fermenters, for example, Salmonella, Shigella, Yersinia. Now, this is a flow chart which shows the classification of Enterobacteriaceae according to lactose fermentation growth on Macunkie agar. Here, Enterobacteriaceae are basically classified into two, that is lactose fermenters and non-lactose fermenters. In lactose fermenters, a sufficient acid is produced along with neutral red dye which is used for staining, therefore pink colonies are formed. For example, bacteria like Escherichia, Klebsiella, Enterobacter and Cytobacter. Whereas in lactose non-fermenters, no acid is produced here. Therefore, the colonies are colorless. For example, bacteria like Salmonella, Shigella, Proteus and Yersinia. Now this picture shows growth of Enterobacteriaceae on Macunkie agar. At first, here is the uninoculated plate. In the second plate, due to the presence of non-lactose fermenters are like Salmonella, Shigella, Proteus, form colorless colonies in the plate. In the third plate, due to the presence of lactose fermenters are like E. coli, Citrobacter, Klebsiella, Enterobacter, form pink colonies in the plate. Now, fermentation by Enterobacteriaceae. Fermentation by Enterobacter species is more limited when compared to some Klebsiella species. For example, Enterobacter produce gas from Cellobiose. Here, Cellobiose is a disaccharide. It is classified as a reducing sugar. And ferment rhamnose, where rhamnose is a naturally occurring deoxy sugar which can be classified as either a methyl pentose or a 6-deoxyhexose, a feature that distinguishes them from most species of Serratia. Now, Enterobacter lactose fermentation. Enterobacteriaceae are often isolated from fecal matter on containing lactose and a pH indicator. Colonies that ferment lactose would produce sufficient acid to cause a color shift in the indicator. As I explained before in the flow chart, in lactose fermenters, where the pink colonies are formed with the production of sufficient acid along with the addition of neutral red dye, which is used for staining.